So here's just a quick review of transcription and translation for protein synthesis. So remember, the first step happens in the nucleus. You have your double-stranded DNA molecule. It has to unwind and untwist and then finally unzip. And you have your mRNA molecule right here that's being formed from it. Notice that there's U's, and that's how you know it's mRNA. Instead of the T's, that's how you know this is DNA. And this is going to keep going until it hits a stop codon. That tells the mRNA molecule it's done and it's ready and it can leave the nucleus. Once the mRNA molecule is transcribed from DNA in the nucleus, it leaves where it finds a ribosome. That's this white thing in the background. Here's our mRNA molecule again. mRNA is read 3 letters at a time. These are called codons, and that's what attracts the correct amino acid, the correct tRNA molecule with the correct amino acid in the right order. On the other end of the tRNA molecule at the bottom is the anticodon. This is going to be complementary to the codon. So this says GUG for the codon, so the anticodon is CAC, and that brings this amino acid here in that right order. This is called a peptide bond that's formed, and this growing amino acid chain is also called a protein or a polypeptide, and it will keep growing until it hits a stop codon. And you can see here's a release protein, and it's going to keep moving down the mRNA molecule, and more tRNAs are going to come in. And then the empty tRNAs or uncharged tRNAs without the amino acids will leave. And this is what you will see in one of your other activities. Now, looking at this on a molecular level, looking just at the letters, the first part, transcription, is what we practiced already. So if you're given a DNA template, remember your complementary bases. And remember, there's no T's in mRNA. So this is transcription. Translation is going to take those same those same codons and transfer that into anticodons. So, and again, you're going to have your complementary bases. So again, there's no T's in RNA. So remember, A and U go together and G and C go together. So that's how the codons and the anticodons match up. And then to make your proteins or your amino acids, you're going to use something called a codon chart. And you're going to take these 3 letters at a time. So we have UAU. There's our first codon. And so if you notice here, that amino acid is tyrosine. The next one's GCC. See if you can figure that one out. So here we have G, C, C, and alanine. And finally, our last example is UCG. So we have U, C, G, and that tells us it's serine. And so this will keep going until it hits stop codon, or in our different lab activities until you make the protein and the traits that you're trying to get for your organism. So what I just showed you here is the interaction that you see right here, your codons and your anticodons. You can find out the right order of the amino acids that way, or sometimes you need to use the codon chart, and that's showing you this interaction with these particular amino acids. And you'll always be given this information. You just have to know how to use it.